the fermentation bioreactor, especially for the gas fermentations. And then that's so interesting. Another topic here is about isolation of gas fermentation microorganisms and their robustness. Probably so practical things. And I don't know, like some professors here, I found some professors here joining this lecture, and probably can ask how to do the isolations for the microorganisms. So you can develop your own microorganism. I mean, like you get it in our country, Indonesia. So that's so interesting. So before Fuad start, probably I would like to share another things here. Think, hold on. This one, I think so. Okay, this is. Can you see? Can you see it? All right.
I can see it.
Okay. Okay. Fuad is a postdoctoral scholar in Pennsylvania State University. Now he actually hold his PhD in Washington State University, and now he's doing postdoc in Pennsylvania State University. So he worked a lot on gas fermentations and then bioreactor and bioinformatics and molecular biology, a lot of things related to microbiology. He did a lot of things. Now I think your project's now a little bit different. You work on the chemical reactions, so that's interesting as well. So, and then Fuad also already published a lot of articles here. If you can see here in 2024, he published like around 4 articles, and in the good journals. And one of them is about biogas upgrading, and then gas fermentation as well. So he also already attended a lot of conference here, and in 2023 we attended the same conference. We went there in SBFC conference. So and this is the technical skill that he has, and then he also already get a lot of awards here and honors. And yeah, I think that's stuff about Fuad. And then it's your turn, Fuad. So you can share your presentations now.
All right. Well, first of all, thanks, Woody. Thanks for the
Yeah,
introduction.
thank
As Woody mentioned, we are friends. We were doing PhD together at Washington State University, and so yeah, it's an honor to me to today be able to share with you a little bit of my research in gas fermentation and research that Woody knows very well, and I'm sure. I mean, I hope you enjoy Woody. Um, Also, I'd like to thank everybody for waking up at 8 or before a.m. in Indonesia. I hope the lecture is interesting to you and it's not boring. I hope you learn something new and um, and I hope it has some impact in perhaps you're doing some research. I'm not sure if there's only undergrads in the audience. There's graduate students. Perhaps you learn something new for your research. And if there is any professor, well, I hope I am standards that you may have. So the title of this presentation is Gas Fermentation Bioreactor, and I would like to have this subtitle as well. And yeah, so how can we use the gas fermentation bioreactor to develop technology that benefit the environment? So first of all, I'd like to talk a little bit about my journey as a researcher. And so, I'm originally from Mexico City, where I studied my bachelor's. And so I was there in Mexico City, doing Mexican stuff, eating tacos, being a good person. And I had a chance to work in a laboratory with Dr. Odin Rodriguez, who was my advisor during my bachelor studies. And so I started working with anaerobic digestion of food waste. This is a photo of me and my advisor and one of my colleagues back then in an anaerobic digestion plant. He looks like he's falling asleep. I think he was just protecting himself from the sun. Later on, I had a chance to go to Spain, to Madrid, and make a very short research stay over there, where, well, I got to travel to Europe, and I work a little bit with algae. I participated in a field campaign to study the physicochemical properties of tubes in the Iberian Peninsula. That was, a different kind of research that I was used to, and it was a very interesting experience. Then I had a chance to study one semester in Poland. Uh, uh, very a very beautiful country. I made so many friends over there, and I had a chance to study computational fluid dynamics, which was very, very interesting and new to me. Later on, I started my PhD in Washington State University, where we have amazing hiking trails. Woody may know that place, Kolchuk. I made so many friends over there, including Booty. Um, Here's me working with microbes and molecular biology. I had the pleasure to meet Luna, the a friend's dog that was very nice and I loved to play with. I work with many different types of bioreactors that I developed in the lab. And finally, I graduated in last May with my PhD in engineering science. And so right now I'm located in Pennsylvania, where I'm doing my postdoc. Work with amazing people over here in conversion of food waste using chemical and biochemical pathways into biofuel and bioproducts. Um, this is a picture. This was a picture of a hiking trail that is around here that I like a lot. I'm sorry, the picture is behind behind the other picture, but this is the first city that I've seen. There is rabbits running out around the city, and that's really nice. And so right now I'm developing pathways for conversion of waste into biodiesel, which is shown here in the picture. And so let's jump into more technical stuff. Today's lecture, I would like to talk about gas fermentation and its application in CO2 upgrading. That's the first topic that I'm gonna talk to you about. And so let's jump right in. So I like to start talking about climate change. First of all, I think we need to state that climate change is real and it's here to stay. Right now, I are you guys? Oh, I think my PowerPoint crashed.
Yeah,
Let me
it's
open
different.
the presentation
Yeah.
again. Are you guys looking at my screen right now or not?
Yes. We are looking
Am I
at yours,
sharing the screen?
but
Okay,
right.
here it goes again.
Okay, that's the first one.
All right, sorry about that.
Yeah, that's the first slide, I think.
I have to go through all of them.
All right.
Okay,
Yeah, there you
all
go.
right. So as I was saying, climate change is real and it's here to stay. There's already proven effects that we are able to measure and to quantify. Some of them are floods and drought, livestock mortality, increase of food insecurity, damage to infrastructure, and larger and faster spread of disease. For example, malaria, which is a disease that is transported by mosquito. As temperature rises, mosquito breathing it's a little faster, and that also increases the rate at what malaria is spread around. So there is many different, very complex effects that climate change is already having on human populations and ecosystems. In 2024, sorry, 2023 was the first year in which we already reached a plus 1.5 degrees Celsius in temperature. So this is real and it's a big problem for us. It's actually, I would say, the defining issue of our times. So one of the solutions to this, well, perhaps one of the main actors in the climate change problem is CO2. So CO2 is emitted by anthropogenic emission sources, and especially by burning fossil fuels. It accumulates in the atmosphere, and this is what retains energy from the sun, and that's what causes the temperature increase. So one of the solutions is to capture this CO2 and convert it into something useful. Um, a little bit of statistics. We know that the natural gas industry, at least in the US, it's very big. It's one of the main primary energy sources for electricity generation. And the trends, the projections, they show that natural gas is going to still be one of the main sources of energy, no matter what, at least till 2050. So we need a solution in order to both capture CO2 and produce renewable natural gas. And so one way to do that is using microbes and using gas fermentation. And so these are two reactions that are very well characterized for gas fermentation. The first one is conducted by hydrogenotrophic methanogens, for example, this Methanobacterium thermoautotrophicus. And so in this biochemical process, CO2 is used as a carbon source, hydrogen is used as electron donor, and this microbe produces methane and water. There is another sort of version of this same reaction, which is conducted by homoacidogens like Borrelia thermoacetica. And in this case, the final product is acetate or acetic acid and water. The only difference really is that during homoacetogenesis, 2 moles of CO2 are consumed per every 4 moles of hydrogen. So at least from an environmental point of view, perhaps homoacetogenesis is a little more beneficial than methanogenesis, under the condition that you find something useful to do with the acetic acid. These are the names of the biochemical pathways that these microbes have in their metabolic machinery in order to make this process to happen. So now, how can we apply gas fermentation in order to benefit the environment? This is a nice picture trying to show that. What we can do is to take carbon dioxide from many different emission sources. Here during my PhD, I work with biogas, which is a product of the anaerobic fermentation of different organic substrates, like for example, wastewater or solid, like municipal solid waste, perhaps also biomass feedstocks. And this anaerobic digestion process produces some methane, but like 40% of the biogas is still CO2, which can be separated and it can be introduced in a gas fermentation bioreactor mixed with hydrogen that is produced from electrolysis. And then depending on what microbe you have in this reactor, you are able to produce either methane or well, and I was supposed to show here as well, or acetic acid if you had homoacidogens. My PhD was focused on methane production, so this is probably what I'm going to talk to you about the most. And so what are the benefits of doing this? First of all, of course, is the CO2 capture. Then you obtain emissions offset by replacing the use of fossil fuel, because you can use this methane to replace fossil natural gas, and in that way prevent the renewable emissions of CO2. Of course, as this can benefit or can bring economic benefits to the wastewater industry, it could increase the feasibility of waste management. And this is technology that can be applied to power to gas projects, which consist on transforming electricity into gas. Once again, the source of hydrogen, or at least what we propose to do, and many different authors in the literature propose to do, is to use hydrogen produced from electrolysis that is powered from renewable electricity. It could be, wind, hydroelectric, or sun energy, photovoltaic energy, basically. And so I talked to you about anaerobic digestion and biogas plants that if we want to look a little deeper into it, so what are these? What are these facilities? So a biogas plant typically uses a substrate, either wastewater or an organic solid waste. Depending on the properties of this waste, it perhaps needs to go through a pre-treatment. From this pre-treatment, you try to increase the putrescibility of the substrate so that whenever it enters the anaerobic digester, it's easier for the microbes to consume and transform into biogas. So on one side, you have the biogas coming out from the digester, and all the solids that were not converted into biogas are called digestate. The digestate side can be dewatered, it means removing water from it, and then you can valorize those solids as fertilizer or soil amenders. They can be applied to soil to increase the productivity of soil. Um, and on the other side, the biogas, as I kind of mentioned previously, it is around 60%, it's composed of around 60% methane and 40% CO2. So ideally, we want to remove CO2 and other impurities in order to produce a almost pure methane stream that can be used in energetic applications. This is a picture of one of the biggest biogas plants in Mexico. This is how digesters typically look like. They are continuously stirred tank reactors. Um, and so, yeah, as I said, after biogas upgrading, you should have a biogas stream with more than 95%. And this is not called biogas anymore. This is called renewable natural gas. And so the energetic applications um, that we can power using the upgraded biogas are many. These include heat, electricity, combined heat and power. You can export the renewable natural gas using existing pipelines, and companies can sell this as biofuel, essentially. It can also be used as vehicular fuel. Uh, in some small communities, it could be used as for cooking, for drying, for cooling or refrigeration, and also to power, for example, pumps. And it could be used to generate mechanical energy. So really, basically, any energetic application that you can think of could be powered by uh, upgraded biogas. And so here's where gas fermentation plays an important role, which is in this stage in biogas upgrading. What we have been trying to achieve is to convert the CO2 that is in biogas into more methane. Um, so why biogas plants are so attractive to us is so we have more than 2,000 biogas plants in the US, and unfortunately, only 5% of them actually produce renewable natural gas. Most of them, what they do is they obtain the benefit of decreasing the amount of solids that they have to treat. They perhaps use them as they perhaps obtain the digestate and they obtain the benefit of using it as a fertilizer, but they don't have enough money to actually build the infrastructure they need to take advantage of the biogas. So what they do is to just burn it, because as probably some of you already know, methane is, I think, like 25 times more powerful in terms of global warming potential than CO2. So we cannot just release it to the atmosphere. Biogas plants need to at least burn it, flare it. Um, and to my eyes, this is like a waste of resources. Like this is energy that they are not really taking advantage of. Um, so by developing gas fermentation technology, we could eventually push the field in such a way that the industry is able to actually upgrade the biogas and take advantage of it. Um, so I have a couple examples of success in terms of biogas and biogas upgrading. The first example I want to show you is um, the municipal solid waste digesters that are located in Sweden. They are used to power the public transportation system. And so they have these pumps in the street where I believe cars still cannot use it, but the bus stops by these pumps and they take they upgraded biogas or just to use it as fuel. And I think it's 100% powered by biogas. And this is a video I took in Malmö, where these green buses are the ones that are powered with like 100% biogas. And it's a really nice city. I think there's a part where I show the turning torso, which is a really nice piece of architecture, just as a curiosity. And so another, I will say, very interesting example of success of using anaerobic digestion and biogas upgrading to recover resources is this plant located in Mexico, which uses nopal waste. Nopal is a plant that is very common in Mexico. It's like a cactus, and so there are parts of the plant that are not really good for human consumption, and so they retreat this nopal waste and then they put it inside a anaerobic digester, which is shown in here. And then they produce the biogas, they upgrade it, and they use the biogas for combined heat and power, which is used for in situ heating. They produce heat with the biogas and heat up the anaerobic digester. And I think they also export electricity. Uh, they are connected to the grid and they export electricity. And they have to work with the producers in order to be able to obtain the nopal for their plant. And so in many ways, they are integrating many different parts of society by having this plant in there. I think it was installed in 2018. I'm not sure if it's operating right now, but I hope it is. So if gas fermentation is such a great opportunity for us to recover or to increase the methane production from biogas or really to convert any CO2 stream into methane, so why is it not widely applied all over the world? So there are three limitations that we've identified that are the ones preventing the technology to advance forward. And so the first of all is that as the process requires hydrogen, it's electron donor, and hydrogen is very less soluble in water than CO2. We have mass transport limitations of hydrogen that limits the reaction rate of the process. Another problem is that often we find that there is a very inefficient use of hydrogen by the microbes that catalyze the reaction. So we need to overcome this problem as well. And on the other side is that we know there's microbes that use CO2 and hydrogen to produce methane, but the cost of cultivation of those microbes are either unknown, non-well characterized, or they are very high because often they require organic growth factors to be cultivated. So these are three challenges that I tried to tackle during my PhD, and I'm going to tell you more about them next. Um, Something else that is interesting to think about is about robustness. So when you have a biomanufacturing objective and you want to produce something using microbes, there's sometimes this problem that you have a microbe, you have a strain that is like this one. It's a little bit of a brat, and so it's like he's saying, my enzymes get cranky every time you change the temperature, and then it comes out like it comes in contact with oxygen. It's like oxygen. I need a vacation. Like it's a very weak microbe that doesn't like any changes in the conditions. It needs to grow in very specific culture conditions. And so what would you prefer? Like this brat microbe, or would you prefer one that says I ferment like a champ, no matter what you throw at me, or I'm here to fix your CO2 problems? Like perhaps we want this, no? Like perhaps it sounds a better idea to work with a microbe that is not as picky. Um, and so part of my PhD was also focused on not just producing this microbe, but also trying to understand what makes a microbe, like a powerful microbe, powerful. um, so yeah, one of the big question is like, how do we get from here to here? So I'm gonna also try to show you a little bit about that. I like to start Well, I mean, next, I'd like to continue talking about the mass transfer limitations of hydrogen and to tell you about some strategies that we could use in order to overcome these problems. Um, I know there's plenty of time, but I'm trying to finish in 1 hour. And
Oh, yeah,
I don't
you
know
still have
what
a
do you
good times now.
say again?
Yeah. Yeah, you still have a lot of times, like more
All right,
than 1 hour. Yeah.
all right, all right. I just like to probably I would like to stop here and ask if there is any comments, any question, or really any insights about the previous ideas that I just showed you. I think I'm gonna do these stops in every section,
Yeah,
so
for a second.
it's a little easier to answer questions.
So is there any questions here you want to ask to Fuad?
All right, all right. I'll keep going, and
Yeah.
but still feel free to ask me like questions about the previous section at any time. No problem.
Mhm, mhm.
All right. So this is an overview of the strategies that we could use to overcome these problems of mass transport limitations. As a summary, we have these 5 strategies. The first one is selecting the right bioreactor type. The second one is increasing the pressure of the system. This, of course, increases the solubility of the gases. Just think about Henry's law, for example. Um, Also, there is a phenomenon called the direct transport into biofilms. I'm going to explain you in more detail in a second. There's also mechanisms for bioelectrosynthesis and also for nanobubbles application. These two are the least studied. And so I'm going to focus more on the experimental evidences that support the use of these three strategies, in terms of overcoming the mass transport limitations of hydrogen. All right, so perhaps there's someone asking, like, what is exactly is this problem of the mass transport limitation? So let's talk about that. Let's picture a system where we have a microbe which is swimming in liquid medium. So we have a liquid phase on one side, and on the other one, we have a gas phase where the hydrogen is. And the microbe wants the hydrogen. So the hydrogen needs to dissolve, reach to the microbe, and then different enzymes in the microbe will convert this hydrogen into, let's say, power for conducting the reactions. Uh, in this case, methanogenesis. No. So if you notice, there's a lower abundance of hydrogen molecules in the liquid phase when compared to the gas phase. And the reason for this is because the solubility of hydrogen is very low. This is due to the poor interaction or a weak interaction between hydrogen and water, which is something we cannot change really. Like that, we have to live with that. But for instance, CO2 is, I think it's 25 times more soluble than hydrogen. So really the limiting factor is hydrogen solubilization in this system. So if you remember your mass transport phenomena classes, you will perhaps remember this equation, in which what we are showing is that the reaction rate is in equilibrium with the mass flux of hydrogen in this system. It means that as soon as the hydrogen gets um, into the liquid phase, it is consumed by the microbe, or at least the rates of those two phenomena are equal. That's called equilibrium or steady state. And this reaction rate, it has a driving force, which is the difference in concentration of hydrogen, in the gas phase. And, this E, it's the concentration of hydrogen in equilibrium, which could be calculated using Henry's law, for example. This is very related to solubility of hydrogen. And then we have this global mass transfer coefficient that accounts for all the mass transport um, resistances, all the resistance to mass transport. And this could include many, many things. And it depends on uh, many, many variables that are associated to phase velocity, uh, temperature, partial pressure of the hydrogen. Also, if you have more biomass concentration, you expect a higher reaction rate, and perhaps, and I mean, for sure, you'll have a different mass transfer coefficient. And also, for example, concentrations of salts and other solids can also alter this. Uh, we call it a constant, but it depends on many, many things, and it ends up being actually a variable. But in a steady state system, it's a constant. It's very common, KLA, the global mass transfer coefficient. And so, sorry, so any strategies that we find that could increase the the mass transport and can impact KLA, that can make KLA more positive, are actually strategies that we can use um, to overcome the mass transport limitations. So, as I said, one of the first strategies that we have is selecting the right bioreactor configuration. If you think about the typical continuously stirred reactor, it has a very poor gas liquid mass transport. So perhaps this is the worst reactor type that you can use. Then you have the trickle bed bioreactor. This one has the highest KLA and hydrogen uptake. The disadvantage is that it is harder to construct because you need to have a packing material inside. You need to have also liquid recirculation, and also it's hard to model. A CSTR is the easiest thing to model in terms of reactors, but trickle bed, making assumptions and satisfying those assumptions is really, really tough. So modeling this reactor and then, for example, sizing it and constructing it in large size, it's kind of challenging. Um, and so the reason why the trickle bed bioreactor has a higher KLA is just picture this. You have liquid where the microbe is growing, you are recirculating it and spraying it on top. This is a packing material, usually very porous. And what happens is that the liquid forms a very thin film, um, which allows for um, having a very high contact area, contact surface between the two phases, between the gas and the liquid phase. And that's the reason why it has a higher KLA than any other uh, bioreactor um, for gas fermentation. And then there is a third type, which is very promising. By the way, here I'm showing the KLAs based on literature. And this is also the some of the experimental hydrogen uptake rates in millimole of hydrogen per liter per hour. And so, as I was saying, the third type of reactor is the membrane bioreactor. This reactor consists of a liquid phase flowing on the outside of a membrane, and you have then the gas phase running inside the membrane. So is similar in principle to the trickle bed in terms that it increases the surface area for mass transport, but it still is not as efficient in order to overcome the mass transport limitations. Um, I think many research is required in order to characterize the membrane bioreactors better. I know um, changing the material of the membrane and also changing the flows can dramatically change the KLA. So I believe there's still a huge need for research in terms of membrane bioreactors. Um, However, from my point of view, having the membrane will always add extra resistance to the mass transport when compared to the trickle bed. So my feeling is that the membrane bioreactor perhaps will never reach the efficiency of a trickle bed. Um, so knowing that, I believe trickle bed bioreactor is the preferred type of bioreactor. There are other things to consider. For example, doing bioelectrosynthesis in a trickle bed bioreactor is not easy. I would say doing it in the CSTR could be even better. Uh, but still, if we just conduct normal gas fermentation, trickle bed is the winner. So now let's talk about pressure increase. Once again, think about Henry's law and also think about Boyle law. Um, Henry's law tells us that the higher the partial pressure of the gas in the gas phase, the more gas will be able to solubilize. So that's kind of like an obvious strategy. If we increase the pressure, we increase the concentration of hydrogen in the liquid phase, and then perhaps will increase the biomass growth rate and the biomass concentration, have a higher conversion rate then. And yeah, the experimental evidence is also very clear, both for methanogenic and acetogenic gas fermentation. We see that the more we increase the pressure, for example, in this reference, the more we increase pressure, the Parts pressure of hydrogen, we obtain a higher hydrogen conversion efficiency. And on the other side, the higher the pressure, the higher the final product titer using Clostridium lambda. This is kind of like an obvious strategy. However, if we think about Boyle's law, what happens when we increase the pressure is also that we increase the volumetric flow of the gas. So we cannot indefinitely increase the pressure of hydrogen because we run into the risk of having a higher volumetric flow than the microbe can actually, be biologically able to take. So we need to be careful in this sense that we cannot really increase the pressure as much as we want. There is a limit. What the limit is, I think we still don't know, but there is a limit. And also very important, having high pressure in a bioreactor with hydrogen is kind of dangerous. So you also need to think about the safety problems that you may have by managing very high pressures. So pressure increase, I think, is a must in every single gas fermentation bioreactor. I think if they don't depressurize the bioreactor a little, they are committing a mistake that they cannot go very, very high in pressure either. Now, this phenomenon, the direct transport into biofilms. And for this one, I like to think if you have made any culture in Petri dish, I just want you to think about that colony that grows on your agar, which is in direct contact with the air, and it's taking oxygen directly from the air. There is no liquid. There is no liquid phase there. So in the same way, if we mobilize a microbe on a surface, in this case, a gas fermentation microbe, and we decrease let's say the size or the thickness of the liquid phase by a lot, what researchers have found is that the mass transport of hydrogen into the biofilm, into this immobilized biofilm, becomes independent from KLA, from the global mass transport coefficient, and it becomes more dependent in the gas diffusivity. What this is telling us is that the hydrogen is kind of like directly being uptaken from the gas phase by the microbe. Now, this is, I would say, an elusive phenomenon because it's very hard to study systematically. This is based on experimentally informed simulation. So there's still a lot to study about it. But it's still based on those, on some evidences that show that having a less wetted carrier material in trickle bed bioreactors lead to a slightly higher hydrogen utilization rate. So there is a big chance that this is actually real, that this is an actual strategy we can use to increase the utilization of hydrogen. So these and the other two strategies are more broadly discussed in a recent paper from me and my P advisor that you can find in Bioresource Technology. I mean, if you're interested, I'll be very happy to send you this paper. Now, I'd like to stop here once again and ask if there is any question.
So is there any questions from the audience here? Mm-hmm.
Okay.
You can continue.
All right. I'll keep going. All right. So next thing I'm going to talk about is the process engineering of a lab scale gas fermentation bioreactor. Here, if you guys have taken any class on instrumentation, I think you like this part of the lecture. So this is a pipe set instrumentation diagram of a biomethanation system that I built in the lab that I was working at Washington State University. And so the core of this system is a trickle bed bioreactor. This reactor has a liquid recirculation from bottom to top. The liquid is sprayed on top of the bed. This reactor had a bed made of crushed clay pebbles. On the other side, we have also gas circulation from top to bottom. There is a system of check valves to prevent backflow from the gas circulation line to the gas inlet line. We had a gas filter to avoid or to minimize microbial contamination. The substrate we used for this reactor was actual biogas sampled from a biogas plant. So that gas can definitely have some microbes in it, and therefore there was a need to have this filter. We wanted to have a constant pressure inside the headspace of this reactor, so we added a pressure switch that was connected in loop with a solenoid valve. And even though it's not directly shown in the diagram, there was a pressure sensor, which was part of the pressure switch that was installed in the headspace of the reactor. So in that way, every time the pressure decreased below the set point, this pressure switch will close the circuit, open the solenoid valve, allow some gas to go to this gas line, pressurize the reactor again, and then it will close the solenoid valve again. So, so in this way, with this mechanism, very simple, we were able to maintain the pressure in the reactor constant. What else we have? So we have a temperature controller. This is a temperature transmitter, temperature controller. We had a manometer. On this part of the reactor, we had a heating belt to maintain the temperature in the reactor at 55 degrees Celsius. And I think that's the main instruments and parts of this system. On the liquid line, we had the broth, which was the kind of like the spent liquid from the bioreactor. And on the other side, we had fresh medium that was used to feed new nutrients to the bioreactor. This is a picture of the system. It's kind of like an old picture because I remember it had more stuff in it, but this is a good illustration of it. We tested that it could actually control the temperature. We tested for no leaks. This was a leak-free environment, and it was built inside a fume hood to prevent any potential explosion from leaks. This is the material that we used to pack the reactor. We fed the biogas into this tank using a aluminum gas bag that was containing biogas, and we used industrial grade hydrogen to provide hydrogen. And of course, we made the gas blend in such a way that we had 1 mole of CO2 per 4 moles of hydrogen. Here you can see the filter that I told you about. This is the pressure switch. This is a valve that we use to control the volumetric flow of gas coming out from the reactor. This was a sampling port. And so, very important, we measured various parameters in the gas inlet and the gas outlet, starting from volumetric flow, but also we determine the concentration of methane, hydrogen, and CO2 using a mass spectrometer. And we use an electrochemical sensor to detect the hydrogen sulfide. As you may already know, hydrogen sulfide is a pollutant in biogas that we were interested in monitoring our experiments. About the liquid outlet, we measure pH, dissolved hydrogen. We used a spectrophotometer to measure optical density. This is a measurement that is proportional to the concentration of biomass in the liquid. And we use HPLC to measure volatile fatty acids production. This was inoculated with a methanogen, so we were not expecting any BFA. However, if we detected BFA, it meant that we had contamination. So we monitored this parameter as well. These are more specific experimental conditions, but in a nutshell, what we were trying to do was um, to test if using real biogas will impact the performance of the bioreactor. In the beginning of the experiment, we used a mimic. This was like kind of like an artificial uh, biogas that we bought from a gas supplier. And when we reached steady state and we established a performance baseline, then we switched this biogas, like this kind of like biogas substitute with real biogas. Um, and so in that way, we tested the effect of actual biogas. And these were the results. Here, blue dots are methane, red dots are hydrogen, and green dots are CO2. This is the composition of these compounds. And also, as I said, we monitor the concentration of biomass and the pH. Here, this vertical line marks the time point in which we replace the mimic biogas with real biogas. And so what we observed is that there was no significant difference in biogas upgrading performance in between these two time periods. So that was good news. It means that the microbe is able to handle um, a little bit of contamination in the gas feed. Um, in you know in average, what we observed is that there was a 99% hydrogen conversion efficiency, something very similar for CO2. It means that in the outlet we had almost pure methane, and this is great. Like this is awesome. This is exactly what we were looking for, what we wanted. Um, but still, some of the challenges now is like we are operating at a very low flow rate. So we still needed to experiment in order to identify what were the maximum flow rate that we could achieve, like, that we could have in the bioreactor without compromising performance. However, just based on this, and when we compare against other studies, we obtain really good performance. Um, and the very important thing that we notice here is that we did not need to feed more than 4 moles of hydrogen per mole of CO2 to achieve a great performance. When we see other similar studies using mixed cultures and not a pure strain of methanogen, what we see is that at similar hydrogen to CO2 ratios, they obtain, it's not a bad performance, but it still is not the full conversion. And from here, we hypothesize that perhaps there's something in the mixed cultures that causes to have a low um, uh, conversion rate or a poor utilization or slightly more poor utilization of hydrogen. Um, so in order to in order to we were interested on knowing what was the um, what was the maximum attainable reaction rate of the reaction if like what will happen if there was no mass transport limitations? What will be that maximum specific um, rate? So we've conducted another experiment in batch in these little tubes with the same microbe, the same strain, um, and so we exposed the microbe to a very low concentration of hydrogen initial, um, and then we had many replicates of this same tube, and we sacrificed 3 of them every couple hours, and we monitored the concentration of hydrogen. And so here is the hydrogen tension over time in this kinetic study. But the most important thing here is after that, we took this data and we feed it the data to the Monod model. Problem with Monod model, which is a biomass growth model that it allows you to model, for example, the growth rate in terms of the concentration of a substrate in the fermentation. We needed to use a more general model because there is really no way to fit any kind of experimental data of this type to this model. So we use something called the integrated monodel, which is actually two equations, two differential equations. Um, we pick the one that describes the biomass concentration over time, and um, we develop this differential equation that that we can use now, that we can solve in order to fit our data. Now the problem is this is not a linear differential equation, so we need a numerical method to solve it. And as we have more than one constant in the model, um, we needed to use nonlinear regression uh, to fit the experimental model to sorry the experimental data to the model. And I have um, okay, sorry, I saw the chat, but I think it's not a question. And so, yep, um, here it is. So we use MATLAB in order to once again make a loop between nonlinear regression and Runge-Kutta, uh, numerical model to solve the differential equation. And so, in the end, what we obtain is something like this. Here, the dotted line is the integrated monod solution. I think we achieved a pretty good correlation coefficient. What we see is that this model becomes a little not very good in the end of the kinetics, and what we believe is that at that point probably the mass transport limitations were really like significant, and that's why um, the 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 system started to be kind of like completely um, driven by mass transport limitations. So, but still, using this procedure, we're able to calculate the biomass yield, the half saturation constant, and the maximum attainable growth rate of the microbe. And it was exciting because these parameters um, they were very comparable with the literature. But the literature, like there's not just one article that measures all these parameters. Like some articles measure just the yield, some other articles measure just the half saturation constant. Uh, Another article actually from my advisor, it reports the maximum attainable uh, growth rate. And so it was exciting to see that these parameters now that are produced just from one single effort kind of like match with experimental data or report experimental data. And so I'm not quite showing it in here, but we can use this value to calculate the maximum attainable uh, reaction rate from a gas fermentation uh, under the assumption that there is no mass transport limitations. And that result was published in Bioresource Technology. Also, as Budi mentioned, we both together we went to the SBFC conference in Portland, and I presented part of my results in there as a poster. And also, we submitted a patent application for the use of the microbe in biogas upgrading. All right, I'm not sure. I think I have time, but I like to stop in here and ask once again if there is any question.
So ask again, is there any questions here from the audience?
Also, you can tell me like I'm not understanding much. Like, can you explain certain
Yeah,
thing? And I'll be happy to explain in more detail or in more fundamental terms.
yes, you're right.
I'm open.
So if anyone here doesn't understand what talk about here, so is there any? Oh, from Graha Nugra probably can talk here. We can speak up here.
Mhm.
So Graha, are you here?
Okay, so I see his question in the chat. And
Yeah.
so he's saying, yeah, my name is Graha Anugra,
Oh,
and
from
I am from the Agricultural Industrial Technology program in
yeah.
Jaran University. I would like to ask how can the number of cells inside the bioreactor be measured? Additionally, how can we minimize excess media fermentation during the process of scaling up the bioreactor to ensure it remains efficient? All right, these are great questions. And I see
Mhm.
there's a hand. Someone raised the hand. Then
Yes,
let me
Kanza.
answer this
I think you can
this
answer
question.
Graha first, probably. After
All
that,
right,
go to Kanza.
all right. So how can we know? All right. So perhaps this is a good slide to work with. So we used in not just in our lab, but in general in microbiology, we used a spectrophotometer to measure the optical density of a culture. In practice, it looks like introducing this tube inside a instrument that measures the amount of light that is absorbed by the cells in the culture. You need to use a control, a bank to zero the spectrophotometer, and um, usually you use just non-inoculated media in order to do that. And
Mhm.
so that amount of light absorbed is proportional to the amount of cells in the culture. Um, so that I think that answers the first question.
Yes.
I hope.
Probably
If not,
here,
Mhm.
he actually asked about the inside of the bioreactors. I mean, you take the samples from the
Is it?
bioreactors and then yeah.
Yes. I just take a yeah, I just take a sample from the bioreactor from the broth, and I
Mhm.
just measure it in the spectrophotometer. Yeah, sorry, I missed the part that it was inside the bioreactor.
Yeah, yeah.
Uh,
So then the next question is how can we minimize excess media fermentation during the process of scaling up the bioreactor to ensure it remains efficient?
mhm. Mm, so yeah, I guess it comes from the concern of like, what if we are using lots of media to maintain the microbe growing, no?
Mm,
Um, I think it has many answers. Like there might be many answers to that. In our
mm,
case, what I can tell is from the in this reactor, the microbe we use is an anaerobic microbe. It means that
mm,
it doesn't produce many, like too much biomass. It is inefficient in terms of producing biomass, but that's good for us because biomass is not the product. The product is methane. So
mm,
it requires very little nutrients to stay alive and to, stay kind of like growing. So
mm,
in this case, we use ammonium chloride as nitrogen source, some vitamins and some minerals in the media. Um, and so we really just fed the reactor at a rate that the microbe will keep growing. Um, I think a strategy to further minimize the amount of liquid media that we need, perhaps
mm,
is, recirculating the media, not
mm,
like feeding fresh medium every time. That's
mm,
something we did.
mm.
Um, and I think that is pretty much it. I think that's all I can think of. Do you
Okay.
have another insight, Buddy, about
And there
that?
is probably we can calculate it how much needed for the nutrient. Yeah, if you already
Yeah.
have, for example, stoichiometric reactions or I don't know, probably especially for nitrogen source also is important. Then we can calculate it. We can predict it. As I remember in your article also, you made a stoichiometric reactions, right? And then we can predict it how
Mhm.
much and ammonium, for example, required there. So that's
Yeah.
another
Yes,
problem.
exactly.
Yeah, and then we can
That's a
predict
question.
what
That's a
the
good answer.
minimum nitrogen source required for the fermentations.
Mhm.
Yes. So Graha, so yeah, is it clear
Okay,
or you have any following questions? Uh-huh.
okay, thank you to answer Mr. Budi and Mr. Watt.
Mm. Yeah,
You're welcome.
yeah,
You're welcome.
yeah. You're welcome, Graha. And then we have Kansa here. Hi, Kansa.
Yes, Mister.
Yes. So go ahead, yo.
Okay. Good morning, Mr. Fuad and Mr. Budi. First of
Or-
all, I apologize for not being able to turn on my camera. My name is Kanza Putri Kairunisa, and I'm from the Agricultural Industrial Technology program at Pajajaran University. As you have explained about the gas fermentation bioreactor, I would like to ask what are the advantage and disadvantage of gas fermentation bioreactors, And what is the biggest challenge in operating gas fermentation bioreactor efficiently? Thank you, Mr.
Thank you, Kansa. That's interesting questions, I think.
Mhm. All right, all right. So advantages and disadvantages. Mm, I guess we need to think compared to what? Perhaps we something we can compare against is the pure chemical process.
Yes,
For
I think so.
example,
Yeah, submerged
uh,
ferment. Not submerged. I mean, like to just like
for example, the Sabatier process, which is it's a reaction that is catalyzed with metal catalysts, and that perhaps it performs the same. It converts CO2 and hydrogen into methane.
Mm,
So I would say first advantage is the bioprocess. It requires less temperature and less pressure to operate. This process can operate from 55 to I'd say maximum 70 degrees Celsius, the gas fermentation.
mm,
And in the Sabatier reaction, I think it requires like 200 degrees Celsius in like 12 bar to
mm,
operate.
mm,
So I think, in terms of advantages, I think gas fermentation requires less um, demanding, let's say, temperature and pressure conditions.
mm,
Um, in terms of disadvantages, when compared to the chemical process, I would say in the chemical process, I don't think you need anaerobic conditions or like you don't need a strict anaerobic conditions.
mm,
Whereas in this process, if the microbe is anaerobic, so you need to have a sealed reactor. It has to be leak free. It has to, it has to be a bioreactor in many ways. It requires more uh, kind of like you need to take care of it. It's like an alive system. So if you miss a condition that allows life in the bioreactor, you basically screw it up. So I think it's more sensitive, the gas fermentation when compared to chemical process.
mm.
Um, that will be my comparison. What do you think, Woody?
Yes, yeah, that's correct. I mean, a lot of challenges, especially for the gas fermentation that we've deal with. I think especially for the leak. I don't know, like actually still have the problem with the leaks there. If you don't set it up the bioreactor properly, and then probably when you want to make like the mass balance or and if there is the leak, it's gonna be problem. So I think, and then the safety as well. Probably for the other, for example, like it's not gas fermentation bioreactor. It's just like the liquid fermentations. I don't know, like it's not gas fermentations. For that one, it's fine. For the gas fermentation, we need to make sure that the bioreactor is safe. I mean, like the safety things there is we need to consider too. So because we pressurize the bioreactors and then I think it happened to you, right, Fuad, when you set it up the bioreactor first time, you use the
Mhm.
glass bioreactor and then it's
Yeah,
exploded.
and
Yeah, and then you
yeah,
change it.
yeah.
Yeah, you change it to a stainless steel
Uh-huh.
bioreactor. So yeah,
Mhm,
so I think yeah we need to consider it too, especially for the safety.
mhm, mhm.
Yes. So how's I forgot Kanza? The answer is it clear or you have another question?
Yes, Mr. Dan, thank you very much.
Thank you,
You're welcome.
Kanza. You're welcome. We have another one here, Giza. Hello, Giza.
Yes, sir.
Yeah.
Yeah. Thanks for the opportunity, sir. So my name is Giza, and I'm from Agriculture Industrial Technology at Pajajaran University. So I would like to ask about the biomethanation process system. How long the process takes, and what restriction must be avoided to avoid a failure, like anything that could cause a failure. Thank you, Mr.
Mhm.
Yeah.
Yeah,
Thank you.
that's a great question. So, the length. First, in this experiment, it took about 60 hours for the startup, which
Mm,
is really not much. Like it's a couple days. Um, and then from there, we operated, I believe, up to 200 hours. And I had another experiment that went for I would say a couple months, and it was still running with good performance. Um, so the process, in terms of starting up, I think 60 hours is what I've measured, and it's really good. Like I think a bioreactor could that's normal. It's just a normal bioreactor. Um, and in terms of retention time, like if maybe the question is more about like how long it takes, but in terms of retention time, I believe the empty bed retention time in this experiment, um, it's not shown in here, but I if I remember, oh, here it is. It was 12 hours, 12 hours of retention time.
Mhm.
Of course, these conditions the volume of the bioreactor. Now, conditions or events that could mm, make the gas station to fail. I think as Budi mentioned is mm, yeah, I would say leaks, but I would say in leakages, which means that you have oxygen going or air going into the reactor in case you
Mhm.
are operating at atmospheric pressure. You need to use proper materials that are leak-free. In this case,
Yeah.
we use something tri-clamp fittings
Mhm.
and also compression fittings, and
Mhm.
those are gas tight. I mean, if we tie them correctly, we expect at least that there's not going to be any leak either from
Mhm.
outside or from inside.
Mhm.
Um, so that was perhaps the most important problem that we had to troubleshoot. Something else that could go wrong, and that is tricky to figure out at the beginning is the pH. As we are working with CO2, the CO2 is acidic, it reacts with water, forms carbonic acid, and
Mhm.
liberates protons. Carbonic acid is unstable, it dissociates, and it liberates protons. So it decreases the pH of the medium. And
Mhm.
so, um, in order to avoid that decrease in pH, we need to either have a very high conversion rate so that as long as as soon as CO2 dissolves in the liquid, it's consumed, or have a very good buffer system. In this experiment, we provided the buffer using phosphates, and we've noticed that without the phosphates, the reactor will acidify very quick. So phosphates are not really, in this case, it's not like substrate for the microbe. It's just to maintain the pH
Mhm.
uh, in a level that the microbe can live in. So yep.
Yeah. Thank you for the questions. Is it clear or not? So I forgot the name.
It's clear.
Giza.
Giza.
You're welcome.
Yeah. Okay. So we have
Answer.
another one. Yeah, we have another one, Fuad, here from Muhammad Rafli. So good morning, sir. Let me introduce myself. My name is Muhammad Rafli Febriansyah from Agricultural Industrial Technology Program at Pajar University. In the context of optimizing gas liquid mass transfer in gas fermentation bioreactors, what are the most effective strategies to enhance gas solubility and ensure efficient microbial utilization, particularly when working with low solubility gases like hydrogen or carbon dioxide? Are there any recent advancements in bioreactor design or operation that address these challenges? I think you already mentioned it in the PowerPoint. Probably you can explain it again.
Yeah, sure. So in
Yeah,
terms of the strategies,
yeah,
so we can go back a little bit. And so I would say the most important, more efficient, and almost
yeah.
necessary strategy to kind of overcome this low solubility is increasing the pressure. If you think
Mhm.
of Henry's law, you will remember that the gas solubility is proportional to the partial pressure of the gas in the headspace. So that
Mhm.
allows for more hydrogen dissolved in the liquid, more microbe, more reaction rate.
Mhm.
Um, another very important thing to consider is using the right reactor configuration.
Yeah.
Having this type of reactor doesn't let you like it has a very low surface area for mass transport. So either using trickle bed or membrane bioreactor is preferable.
Mhm.
And in the literature, they also report having a higher mass transfer coefficient for these two types of reactors. Um, and I would say those two, like bioreactor configuration and pressure increase, are the most at least more proven strategies to overcome or to enhance the gas solubility.
Yeah.
Maybe I would like to correct there, like gas solubility is a kind of like a constant that depends in pressure. So by selecting a different type of reactor, you don't really change the solubility, you just increase the KLA, the mass transfer coefficient.
Yeah.
Are there any recent advancements in bioreactor design and operation that are these challenges? I would say this is it. I think this is it.
Yeah.
Mhm. I see
So
another question from
yeah, we have another
Timotheus.
question.
Mhm,
I think before Timotheus, we have Albinia first.
mhm, mhm.
Albinia, are you here?
Yes, Mr.
Yeah, go ahead,
Okay.
Albinia.
Good morning, sir. Let me introduce myself. My name is Albinia Trianora from Agricultural Industrial Technology Program at Pajajaran University. I want to ask about how to monitor and control the process takes place in a gas fermentation bioreactor to ensure optimal results. Thank you, sir.
Mhm.
Thank you.
Yes,
Thank you.
so thank you, Albinia. So we can perhaps go through a little bit of the instruments that we use to control this process. I would say this is my design. Perhaps in the industry there are other perhaps even more efficient ways to control the process, but I would say as long as you are able to control the pressure, and in my case it was to maintaining constant pressure, um, I think you are good. And in this system, we used a pressure switch, which was sensing the pressure in the reactor all the time, and opening and operating this solenoid valve to release gas from the gas storage cylinder into the reactor. What this does is to maintaining gas flow of fresh substrate with fresh substrates going into the reactor at all times. It keeps the microbe growing, active in the reactor, and at the same time, it allows for the processed gas to go out in a continuous. I also mentioned about controlling pH. That's very important. Using a buffer solution. And definitely temperature is important. These microbes are thermophilic, so they require to have around 55 degrees Celsius of temperature. So temperature control is very important. As Buddy mentioned, having enough nutrients, which you can definitely calculate, or at least model. It's important. How to maintain those is just, I guess, having a pump with the right flow rate. Also, you might be concerned about contamination with other microbes. So in this case, we had a filter. But there may be, in an industrial setting, there may be other kind of like sterilization methods to secure that you don't get too much microbes going into the reactor. And perhaps another important thing is to keep it flowing. Like if you stop the flow, you stop the substrate income. The substrate going into the reactor for a while, the microbes will start dying. They have this phenomenon called an autophagy, and so they start eating each other, and you don't want that, and you want to keep the substrate going. And I think that's it. That's it. All right.
So how's the answer? Albinia?
Clear, sir. Thank you.
Okay, thank you,
You're welcome.
Albinia. So we still have several questions. I think three, four. Okay, four questions. What do you still want to continue presentations, or you still have
I have one more section. Yes.
Okay, I think you can present first, and then we can go continue the Q&A after that.
All right. Sounds good. Sounds good.
Okay.
All right. All right. All right. Let's see. So this next section is about isolating gas fermentation microorganisms and how to study their robustness, and also what robustness is, which is kind of like a new concept in microbiology. So this is the pipeline that I followed to isolate my own microbe. I started from a digestate that was collected from a thermophilic anaerobic digester that we had in the lab. I used serial dilution in a media that contained only ammonium chloride as nitrogen source. I provided hydrogen and CO2 to the headspace of these tubes. The tubes were sealed using rubber stoppers. The tubes were cultivated, of course, in anaerobic conditions. They were flushed, so there was no oxygen. And also we added reducing agents like sodium sulfide and cysteine in order to decrease the redox potential of the medium. Then, in order to avoid the growth of anaerobic bacteria, specifically homobacillogens, we provided antibiotics. We incubated these tubes for about 15 days until we observed growth in the tube with higher dilution. Later, we cultivated in solid medium in these systems called the roll tubes, which have agar attached to the walls of the inside of the tube. And then we picked one of the colonies in here, and we analyzed its 16S rRNA gene to identify the strain. We also mapped the whole genome, and we conducted just morphology characterization using microscopy. And also we propagated one of these colonies, and we call it a new wild type. And we use this microbe in several gas fermentation experiments, like the one that I just showed you before. And we also try to optimize a little bit the cultivation conditions, or if not optimize, at least to find the optimum conditions for growth of this microbe. This is the identity of the microbe. You can see it looks like hair on the microscope. It doesn't look like nice like bacterial cells. In this case, this is archaea, and it's I think it has a very particular morphology that I haven't seen before I did this project. These are the colonies. Once again, this is archaeal colonies, and this is the only picture I've seen of archaeal colonies. I'm sure they all grow like colonies, but they don't they don't publish how the colonies look like. This is the position of the new strain in the phylogenetic tree of the Methanothermobacter genus. This is our strain, and we call it strain vessel because it was cited in the Bioproducts Sciences and Engineering lab. Then we measured the effect of yeast extract. Also, we measured the effect of using wastewater instead of mineral medium. And we measure also the effect of ammonium chloride on the strain. What we know so far is that there is not a significant difference in the growth rate of the microbe when we cultivate it in wastewater when compared to the basic medium. This is good news. It means we can use wastewater as alternative medium to cultivate the microbe. On the other side, the additives, like when we tested the effect of yeast extract, it seems that yeast extract actually decreases the growth rate of the microbe, which was something unexpected because microbes usually like to have yeast extract, but in this case, it seems that that's not the case, and it perhaps has to do with the chelating effects of yeast extract, which it means that it can kind of like take metal ions, and then those metal ions become less available for the microbe to leave. This is the effect of temperature and pH. We see that the maximum temperature, sorry, the optimal temperature is around 64 degrees Celsius, and the optimum pH is around 8.2. When we compare the growth rate that we obtain in the optimum conditions with this new strain versus the type strain that was the first strain of the of the species that was isolated, we see that it's significantly higher. And we achieved this in a medium with no yeast extract, with no organic supplements apart from vitamins. And that's really good. It means that the microbe is less expensive to be cultivated. This is very molecular biology specific, but we follow this procedure in order to map the whole genome of the microbe. I like you to think about the dogma of molecular biology. We know the genome, then we know all the proteins that the microbe can synthesize, and we know all the functions that it can perform. So this is very valuable in order to understand why this microbe is able to grow without organic supplements, and other microbes don't. These are tools that I use for bioinformatics. This is the map of the whole genome, and very importantly, it compares in size with the type strain. However, what we see is that the hypothetical proteins, this means proteins that their function hasn't been experimentally characterized, and it's just assumed or interpreted from similar proteins, is about 79%. This is huge. This means that we know very little about the microbe, and it's exciting because it shows there's lots of opportunities to work on this microbe and discover perhaps new proteins and perhaps new functions. In terms of phylogenetics, it looks like essentially it's the same as the type, which belongs to the species Wolffi, Methanothermobacter wolffi. However, in terms of functions, in terms of proteins, can synthesize is kind of different. Some of these differences are that the abundance of some of the functions that are encoded in the genome, some of them are more abundant in our strain when compared to the type strain. Some of these functions are protein glycosylation, lysine biosynthesis, oxidative phosphorylation, and nitrogen fixation. This suggests that some of the genes involved in these functions were at least incentivized, or were they became more abundant during the isolation procedure. And this kind of resembles the concept of adaptive laboratory evolution. This specific pathway here is very interesting because it suggests that our microbe is able to synthesize lysine using only hydrogen and CO2, and perhaps the type strain is not able to do that, and that's why it requires yeast extract to grow. There's this small region in here. Here, the length red lines show the similarity of the genes when compared to the type strain, and so we see that this is very low in this region. And some of the genes that we find in this region is ABC transporters, which are related to ATP binding. Also pseudomerane binding. This is related to the cell wall of the microbe. And I mean, other functions that are unknown and that we are not able to suggest on a functional level what is happening there and how different this microbe from the type strain, but at least it suggests, because some of these genes are found in other microbes that are not here, like for example bacteria from the Thermoproteota. I think this is a family, and so this suggests that during the isolation there was also, apart from adaptive laboratory evolution, there was also horizontal transfer from other microbial groups. So we come again to this question about how do we get here? How do we manage to obtain strong, powerful, resilient microbes? And so there comes the concept of robustness. Robustness in microbiology is quantified by the standard deviation of a cellular function when a condition is changed. That means that if this microbe produces methane, and if we see that the methane production is very sensitive to temperature, for example, it means that the microbe is less robust to temperature. If we observe the same when there's small quantities of oxygen, then it means that the microbe it has a low robustness towards oxygen. So based on this intuitive idea, we designed a pipeline, fermental pipeline, to measure the robustness of this microbe um, towards oxygen, temperature, ammonium chloride, and hydrogen starvation changes. And then we use proteomics, which is the study of the proteins that are present in living organism, in order to explain how, in order to explain the observations of robustness, the microbe. So these are some of the results. Here I'm showing a heat map. This heat map is displaying 5 different cellular functions from our methanogen. The scale is a continuous scale from blue to red, where blue means low, means high. And so, for example, if we see a blue in lag phase, it means that it was a short lag phase. It means that the microbe took not very long to start growing. And if we see a red, it means that it took very long to start growing. So, for example, in starvation of hydrogen, we see that having a long starvation kind of triggers the microbe to have a delayed growth. And this other heat map, I'm showing something similar, but in this is the robustness. It is the standard deviation of these cellular functions as a function of the different experimental conditions. And so here, what is not good, what is not convenient is to have low robustness. So low robustness is blue. So we see that high temperatures leads to low robustness. Although certain concentrations of oxygen, like high concentration of oxygen, leads to low robustness, not good. And in here, we are conducting statistical tests in order to see what conditions lead to significantly different robustness for the microbe. And once again, you know, yeah, the oxygen and temperature are the ones that are significantly affecting the growth of the microbe. Now, we have a multivariate data set, so it's appropriate to use a multivariate data analysis tool. And in this case, we use principal component analysis, a PCA, in identify what conditions lead to low robustness, or to kind of like verify what we looked in the heat maps. And so once again, yeah, low robustness, negative robustness in terms of lag phase, it seems to be characterized by low temperatures, while higher changes in robustness in terms of biomass yield, methane yield, and growth rate are more dependent on redox. And so in this case, we are able to kind of like differentiate a little more about like what kind of robustness depends on what kind of conditions. And that's really, really interesting. There's also conditions that do not have much effect on the robustness, and that is also interesting. For example, nitrogen seems to be actually beneficial towards robustness of methane productivity, and that's kind of interesting. Then in what we did next was to all the samples that we generated during these procedures, we conducted proteomics on them. This means that we extracted proteins, and those proteins were analyzed using liquid chromatography and mass tandem mass spectrometry. And these are the steps that I followed for the analysis of those readings for understanding the proteome of the microbe. And so here I'm showing the relative abundance of proteins in different conditions, and we see that at least in this data set, we see that low temperatures lead to significant downregulation of proteins in the microbe, which kind of makes sense because at lower temperature, perhaps the metabolic activity is lower. We discovered many interesting things, but one of them was that some of proteins that are differentially abundant when there is a certain concentration of oxygen in the medium um, are membrane proteins. And very interestingly, one of them it looks like this, like it has this alpha helix that goes through the membrane. And in the outside of the membrane, it has a methionine residue. And this is very interesting because methionine is a reducing agent. So this is like the microbe synthesizing it. So first its own reducing agent, and then synthesizing a protein that it can kind of like deploy the reducing agent and be ready for oxygen attack. So I mean, this is really interesting, and it has never been at least discovered before. So this is a significant advance in trying to understand the response of methanogens to oxygen pollution, contamination. Part of this was published already in Applied and Environmental Microbiology. Also, we uploaded the sequence of rRNA of our microbe, and we also uploaded the whole genome sequence to GenBank. And so this is a roadmap that I followed for my PhD. I think it's valuable to share. I kind of outlined the studies that I performed and how they contributed between each other. I think this is a good exercise for everybody doing graduate studies to have for the thesis, so that you kind of like make sure you have a cohesive piece of work that the committee can see, like what is your progression and kind of like how all the chapters connect to each other. And so as a concluding remark, I would say there's this elephant in the room, which is hydrogen. Hydrogen is very expensive. It's really hard to produce. It's kind of a pain in the neck to be able to transport it and store it. So why would we use hydrogen to produce methane, which is hydrogen itself? It's already a fuel. So I hope some of you are already asking the question, like why would you do that? Why don't you just use the hydrogen and that's it? So there are several reasons for that. One of them is transportation and storage of hydrogen. Hydrogen has a very low density, and therefore it also has a very low power density or heating value. It means that in terms of how much energy you can have in a unit of volume, methane is superior than hydrogen. This makes it easier to transport, easier to store. And perhaps that's the reason why we have so many natural gas pipes and so many, systems for transporting natural gas. We have ships, we have pipes, we have trucks, we have many different ways to transport methane. And we don't have those systems for hydrogen, or at least not yet. So for a while, we will need natural gas and not hydrogen. Related to this is we already have industrial equipment to use methane. We have methane machines. We have combined cycle, huge plants to convert, to extract energy from fossil natural gas. All the combined heat and power devices are, well, not all of them, but there is a few of them that are adapted to biogas or methane. And I don't think there is any for hydrogen. Auto engines that are used a lot in vehicles are also already adapted to methane and not hydrogen. Residential heating and also stoves the burners are designed to burn methane, not hydrogen. So we will need to adapt all these systems in order to use hydrogen and not methane. So those are the advantages of using methane over hydrogen. And so, yeah, I like to give a shout out to my lab mates in Washington State University. Here's Booty present all the time. Booty.
Yeah.
This is our answer.
Always there in the picture.
Actually, these are pictures that Buri. I never take pictures, but Buri is really
Oh,
good at keeping track of them. So these are pictures that Buri gave me at some point. And so all my research during my PhD was funded by DOE, and I'm very grateful for that. And so
oh.
I appreciate your attention, and I'm ready to hear the rest of the questions.
Thank you, Fuad, for the presentation. That's great presentation. I know that your PhD work, right? Most of the things that you present today, it's related to your PhD. So that's great
Mhm.
presentation. But we
Thank you.
actually have a lot of questions. We have 15 more minutes, but we have around seven, I think seven, yeah, seven questions here. So you should answer one by one first, probably. So this is the first
All right,
question
let's give it a try.
from Timotheus. So good morning, Mr. Fuad. Let me introduce myself. My name is Timotheus Andito from Agricultural Industrial Technology Program at Pajang University. So my question is, what are the most common limitation in scaling up gas fermentation processes from lab scale to industrial scale bioreactor? This is about the scale up. So what is the challenges probably here, right?
Mhm.
The most common
Mm,
limitation or the challenges? Okay.
yeah, I think many challenges. Probably I haven't scaled up the gas fermentation reaction yet, or
Mhm.
reactors yet. But I would say having a good um, stoichiometry of it. I think that's the first challenge we have to overcome. I have a stoichiometry for hydrogen and CO2 consumption, but for example, just like Puri mentioned, having nitrogen or phosphorus also for
Yeah. Mhm.
consumption, we don't have those data. We don't have that data.
Yeah.
So uh,
Mhm.
we definitely need that in order to design large scale gas fermentation, and we don't have it. So I think that will be one of the
Yeah.
first things to tackle.
Yeah. And then you also already calculated your KLA, right? And we have to maintain when we
Hmm.
scale it up the from the lab scale to the industrial scale. Probably we can use the KL. KLA must be same because KLA
Uh,
is that related to the mass transfer limitation. So we need to have the same KLA between the lab scale and the industrial scale. I think so, right? For
right, and that's hard to like. It's hard to predict if that's gonna be actually
Mhm.
the case. I don't know if it's gonna be better or worse in terms of mass
Mhm.
transport at the
Yeah.
large scales. So I'm
Mhm.
not really sure, but yeah, definitely that's a challenge as well.
Okay. So how's the answer, Timotheus? Is it good?
It's good, sir. Thank you.
Okay, thank you, Timotheus.
You're welcome.
So we have another Joe Bima Sakti here. Good morning, Mr. Fuad. Let me introduce myself. My name is Joe Bima Sakti from Agricultural Industrial Technology Program. I would ask about the bioreactor design. In the context of gas fermentation, bioreactor design plays a critical role in ensuring efficient gas transfer and microbial activity. Could you explain the challenges associated with scaling up? This is the same question, I think, the scaling up
What is the same
bioreactor
question? Good.
for fermentation, especially in maintaining the optimal gas liquid mass transfer rates and preventing substrate limitation. I think that's the
Mhm.
same.
I think it's the same. Perhaps just to add about the preventing substrate limitations.
Yeah,
Um, in a steady state, the microbe will kind of adapt to what
yeah,
substrate it has available.
yeah.
So yeah, in a steady state, the microbe is not gonna grow further than what it has available to grow on. So
Mhm.
um, yeah, perhaps that is a problem in batch fermentation.
Yeah, yeah, for the fermentation.
Mhm.
So, how's the okay? Probably. So how's the answer, Joby Masakti? Do you have any questions or not?
Oh, sir. Thank you, sir.
Okay, thank you, Joe.
You're welcome.
So we have another one, Martin Simanulang. Good morning. I'm Martin Etan from Pajajaran University. I want to ask about probability. Is it possible if we do a gas fermentation bioreactor only CO2, oxygen, and nitrogen? Because I think these gas are enough. Thank you, sir.
Yeah, that's a great question. Well, I hope he means CO2, hydrogen, and nitrogen. Because
I think
oxygen,
no,
no,
the oxygen is
oxygen.
yeah, yeah. I think hydrogen.
So that's great. That's a great uh thing to think about. Thing to think about.
Mhm.
Because for example, this microbe, it's able to fix nitrogen, or at least that's what we predict from the genome.
Yeah.
So yeah, like if it fixes nitrogen, it will produce its own nitrogen source from nitrogen.
Yeah.
And it's a great idea because sometimes biogas has nitrogen in it that is produced from denitrification of nitrates in the anaerobic digester. So yeah, that's possible. I think it's possible, but it's still to
Mm.
be proven. I think that's
Yeah.
a good experiment.
Okay. All right. So how's the answer, Martin? Is it good? Okay. I think Martin not here. So
Okay.
okay. Yeah. So we go to Gina. Good morning, sir. My name is Gina Mutia Zahra from Agriculture Industrial Technology Program at Penn University. I want to ask how gas fermentation can help the agricultural sector, and is there any research that can prove its
Hmm.
success? Thank you. I think
Hmm.
it's related to agricultural. Yeah. So what
Yeah.
is the competition between gas fermentations and then agricultural sectors here?
Hmm. I think the most obvious connection for me really is once again in anaerobic digestion, because the anaerobic digesters. I'm trying to go back to that slide. They are always related to agriculture, almost always, because many of
Mm,
them are fed with manure or with biomass feedstocks.
mm,
Uh, so if we make anaerobic digestion kind of like more efficient by adding a good biogas upgrading stage, we make agriculture also cleaner because we are
mm,
able to recover more energy from
mm,
the biogas that is produced. Uh, this example here was from using sewage sludge, which I don't think can be considered agriculture, but
mm,
a lot of anaerobic digesters around the world are fed with manure, with agricultural substrates. So yeah, but making gas fermentation or implementing gas fermentation, you indirectly help agriculture. That's probably the most
yeah.
direct impact. And on the other side is if you are able to get more or if you're able to increase the visibility of this part here, perhaps you are also able to you know use the digestate or incentivize the use of digestate in agriculture as fertilizer and soil amenders, and perhaps replace some of the chemical fertilizers.
That's correct. So how's the answer, Gina? Is it good, or you have another question?
Thank you, sir, for the
Oh,
answer.
you're welcome.
Okay. You are welcome, Gina. So now we have Jasmine here. I'm Jasmine, one of the students of Agriculture Industrial Technology from Paju University. I want to ask how big is the role of this technology in the global carbon cycle? I think you already explained it. And the transition to green energy. And what is the role of gas fermentation in supporting a bio-based circular economy? Thank you for the
Mhm.
opportunity. Yeah, it's
Sure. Yeah, that's a good question. So mhm. So in the first part of the question, so what role of this technology in the global
yeah.
carbon cycle?
Mm-hmm.
Yeah, so it will be capturing CO2 from CO2 emissions. This
Mm-hmm.
is a example with biogas, but we can use really any source of capture CO2. So in that way, it's carbon fixation. If we think about like geochemical cycle,
Mm-hmm.
um, it will be the step of carbon fixation.
Mm-hmm.
Um, the transition into green energy, um, you are right. Like it impacts the production of clean biofuel and clean bioenergy. Uh, if we produce methane, that is, in this case, it's renewable. Then yes, like it has certain impact. I think the question is a little bit about how big is it.
Mm-hmm.
So I think that's a great question because I would like to have a number and say, okay, so we can cover a certain percentage of the CO2 emissions, but I think that's still something to solve with research. Um, you perhaps something related to this biogas is considered by the International Panel for Climate Change as a very important and very significant energy source. And so it depends on what's the substrate used for biogas production. But companies can get carbon credits from producing renewable natural gas from biogas. So that definitely impacts the circular bioeconomy in that sense.
Saya udah bilang kan waktu itu saya dapatnya dari TPP. Dia bikin saya
So
suruh usaha. Yeah. So how's the answer, Jasmine? So Fuad?
Yeah, that's enough, sir. Thank you for the answer.
Okay, thank you,
You're welcome.
Jasmine. And we go to another participants here raising hand. I think the first one, I forgot where is that? Who's the raising hand?
I
Widyarti?
think
Uh,
Rayana.
Rayana.
Yes, sir, I'm here.
Okay, yeah.
Okay, good morning, Mr. Budi and Mr. Fuad. So, I'm gonna
Morning.
introduce myself. My name is Rayhana. I am from Agroindustrial Technology 2023 from Pajajaran University. Before that, I would like to apologize for not being able to turn my on camera at the moment. So, for the question is, what challenges are typically encountered during the antibiotic application step? Thank you, sir.
Yeah, I mean, we have been talking a little bit about that in terms of the technology, like the technical aspects that are kind of challenging for implementation of gas fermentation. I think currently a big challenge is that never has really tried to go further and like actually do a industrial sized gas fermentation project. I think there are a few examples in Europe that are kind of like poorly documented. So it's a little hard to tell to this point, what's the state and what have been the challenges for those projects. We have been mostly tackling the research stage at this point, but I would say hydrogen is the biggest issue. For this technology to be feasible, we need to have cheap hydrogen. So if we are not able to produce it in enough quantities at a low enough price, then that is definitely a limitation, and perhaps that's what is limiting the US to transition, and many other countries also into transitioning to using this technology.
Yes, yeah, that's correct. So how's the answer, Raina?
Okay, thank you, Mr. Fuad and Mr.
Thank
Budi.
you.
You're welcome.
So we actually only have 1 more minute for I think so we one last question probably from Widyarti. I think I saw one of them raising hand here. Widyarti. Another one.
Good morning, sir. Let me introduce myself. My name is Widyarti Putri, and I want to ask a question. The question is, what are the key steps involved in isolating microbial strains that's suitable for gas fermentation, and how can their efficiency in utilizing specific gas be assessed? Thank you.
Thank you, Widyarti.
Okay. So I would say first step for isolation will be to use a right inoculum. That means you need to find a inoculum that it contains hydrogenotrophic methanogens or homoacidogens. For that you can choose natural inoculums that could come from, for example, very fancy places like thermal fumes down in the ocean or lake sediments or things that are actually very hard to get. Or you can go the easy way like we did and grab digestate from an anaerobic digester. And there you will find, I mean, if it's a healthy digester, you will find microbes, the microbes you're looking for there. Then next step is to apply the right selective pressure mechanisms. That means you don't want stuff that grows on organic substrates in there. So you want to avoid having any organic supplements or any organic substrates. Organic carbon, organic nitrogen is not good for isolating these microbes because you're going to have much other microbes growing and competing against them. So using the right selective pressure mechanisms. If you're looking for a microbe that uses CO2, then add CO2 to the culture. If you're looking for a microbe that grows on hydrogen, then also add hydrogen to the culture. Then perhaps other selective pressure mechanisms is using antibiotics like we did. This is in case you don't want to isolate homocidogens, but if you want homocidogens, then don't add antibiotics. And then a key step in all isolations is to cultivate in solid medium because you want to have isolated colonies that perhaps came from a single cell. So in that way you have only one cell line or one strain. Yeah, then you propagate this colony and you are set. You have isolated a gas fermentation microorganism. And I think the second question I already forgot about. If you can repeat the second question, I appreciate it.
Oh, I also forgot it. So, Widyarti, what is the second question? But it's related to efficiency, I think, as I remember.
Oh, okay. Yeah, I got you. I remember.
Oh,
So how
efficiency,
to
yeah.
assess the efficiency? Yeah, if you had like many different let's say methanogenic strains and you wanted to screen for efficiency, I would say that's kind of like what we did in our robustness experiment. You want to measure the cellular functions. You need to identify cell functions that you are interested on and that are correlate with the performance of the strain. That means, having growth rate, methane yield, methane productivity, or biomass yield are good variables to measure. And so what I would do if I wanted to compare between strains is measure these variables in an experiment, and as explanatory variable, I will have, instead of cultivation condition, I will have strain. And the strain that is more robust towards certain perturbation or that has the highest growth rate or the highest methane productivity, then I will select that one as the most productive one.
Okay. That's good. That's correct. So, how's the answer, Widyarti? Is it good, or you have another questions?
I get it, sir. Thank you.
Thank you, Widyarti. Thank you. Well, actually, we still have five more questions for it. So I think I suggest everyone's here who asked the questions to email you, and probably you can write it down to your
Yes,
email. Or yeah,
I'm gonna write down my email. You
yes, yes,
please email
and
me any
then
questions. I'll be happy to
yeah,
answer and to chat with
yes, yes.
you guys at any time.
Uh-huh. And I think we also will send you this question to your email, and then you probably can, answer it one by one, and then you send it back to me. Then we will send it to the audience here because we already have their email address.
Absolutely. Okay. All right. All right.
That's a good idea, right?
Sounds good.
Yeah, because the time is over now. Yeah, it's already, yeah. Already 7
All right.
here. I think 10 in the morning in Indonesia and 10 in the evening in your place now.
Uh,
Yeah.
it took very long. I thought it was gonna be quicker than this, but it was. I had fun. I appreciate all
Okay.
the attention.
A lot of
Uh,
questions here. So audience here is so active, I think today.
yeah.
Yeah,
I
yeah.
appreciate your attention.
Yeah,
Attention, guys, and I hope you learned something from this lecture.
sure,
And
sure.
as I said, you can contact me anytime. I'll be happy to chat with anybody of you.
Yeah, sure. So anyway, thank you for the great presentations throughout, and we actually already learned a lot of things from you, especially about the gas fermentations and then gas bioreactors and the applications, and little bits about molecular biology here, and then the modelings little bit. So we learned a lot of things today, and then before we leave from this lecture, I think Aisha, are you still here or not? Aisha?
Yeah,
Yes,
yes. Yeah.
we have the certificate for you here, probably. Can you stop sharing? Okay.
Sure. Stop sharing.
Yeah. So we already prepared something for you. This is the certificate that we already prepared. Yeah, this is certificate.
Thank you very much. Yeah.
And then we will
It
send
has a
Yeah, we will send it to your
well.
email address. Yeah, thank you so much for your effort today because I understand you need to work and you also need to prepare for the presentations today. So that's why this is the things that we can give to you.
It's all right. I
Yeah,
appreciate it. I
yeah,
appreciate it. This is amazing. Thank you,
yeah,
Puri. I
thank you.
like the design. that has a seal. Is that the seal from the university back
Yes,
in
yes,
Indonesia? Okay.
yes, yes.
That's
That
great. I appreciate it.
Yes, that's this is the from our faculty. Faculty means that's different faculty. In the United States, faculty is the professor. In our country, the faculty is kind of like if you have faculty and there are several departments. So
Mhm.
yeah, this is from the faculty, and Dr. Sarifah Nurjana is the dean of the faculties there. So he signed it, the certificate.
I appreciate it. Thank you. Thank you very much.
Okay. Thank you so much for today. Probably we can end this lecture today. And for next week, we'll have one more lecture. And Anggana will give the presentations for next. Yeah, actually, we'll talk about photobioreactor and one more lectures. And yeah, we can continue for the next lecture. Thank you so much. And thank you, Fuad, for today. I enjoy your days there. And then I'm waiting for your wedding invitation.
Absolutely, buddy. Yeah, I'll send you the invitation. And I hope to see you in Mexico, January
Oh,
11th.
okay. Oh, probably you have the Zoom meeting, right? Oh, no.
I might, yeah.
Oh,
We
okay.
could.
Okay.
We'll see, we'll
Great.
see. Thank you for the
Great.
invite, buddy, and I had a good time.
Thank you so much.
Take
Thank
care.
you so much for your time. Okay. Bye-bye. Bye-bye, everyone. Bye-bye.
Bye.